Alright, so this next video is on a process called gel electrophoresis. So now that you know a little bit about DNA, we need to kind of figure out, well, how do we look at DNA? What does that look like? And how do we use it to find things out? So gel electrophoresis is a lab method that we use to separate charged particles like DNA based on the size of the molecule. So DNA has a slight charge, positive or negative, and we can use this technique to separate it out into different segments. So how does it work? Gel electrophoresis is DNA. In that process, DNA is cut using restriction enzymes. So it basically takes that long double helix of DNA, chops it up into different segments. And those restriction enzymes, what they do is they recognize a certain string of letters. So they might recognize A, T, C, G, 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 A. Okay? So that would be the string of nitrogen bases that the restriction enzyme recognizes. At the end of that, after that last A, it makes a cut. And it cuts the DNA into a segment so that we can see that particular segment. And that allows us to kind of see commonalities, things that are the same between DNA segments of different people. But that cut DNA is then placed in an agar, which is kind of the consistency of a Jell-O, kind of, a little more firm than Jell-O. And then it's treated with an electrical impulse. The impulse is applied, and ultimately what happens is all of the DNA segments that are different sizes and weights now, when they're treated with that electrical impulse, they start to move across that agar or that Jell-O-like stuff. And the heavier segments take longer to move down the gel. So you treat this, usually it's a couple hour treatment with gel electrophoresis, and the DNA segments start to move across the gel, and the heavier segments are going to stay closer to where you put the DNA in than the lighter segments. They're going to just move quickly down the band because they're not so heavy. They can move faster. And as the time passes, DNA moves to the other end of the gel. This makes different bands that show the different sizes of the different segments. Because we cut the DNA into little pieces, and each little piece might be a different size, and so they move at different paces. So kind of, this is what it looks like. So right now to you that looks okay, like, okay, that's a bunch of lines on a screen, and some of them are lined up and some of them are really dark. So let's go into what does that mean. So everywhere that the bands line up means the DNA was cut in exactly the same place, because that segment was the exact same size. So DNA is cut exactly the same place. Even more simple, the DNA in those bands are exactly the same. Okay, so we have exactly the same little segments there. So let's break down what our electrophoresis means. So again, what it looks like this. So let's put some circles on it. This first band means that those 4 or 5 individuals have exactly the same DNA sequence in that portion that was cut by our restriction enzymes. At the bottom, these 3 individuals, their bands line up exactly, so their DNA is exactly the same in those segments. And the same with this little guy off to the right. So why is that important? Well, each column of those results that you just saw indicates a different sample of DNA, meaning it might be the sample of mom and dad and baby. In other words, what you're seeing is common genes in 2 different people or from 2 different samples. So this is very helpful in the real world to compare different samples from different sources. So when is it used? A real-life example for the use of electrophoresis is in forensics. You have multiple suspects, all for the same crime, and you found one little piece of DNA on the window that they used to break into the house. Okay? You have that piece of DNA, you run it in electrophoresis, and you have 3 suspects. Well, only one suspect is going to have the bands of their electrophoresis that line up exactly with the bands on the sample you found at the scene of the crime. That's how we do DNA analysis, and that is used in criminal cases to determine individuals that are guilty for a crime. Also, it could be used in, those genetics cases we were talking about where you have an unknown father, a paternity case, and you have done your blood typing and you're not quite sure, you're not going to really put your money on this one guy definitely for sure being the dad. We'll run electrophoresis and compare, and the child's bands are always going to line up with either the mom or the dad. And if they don't line up with the dad, where they're not lining up with the mom, then he can't be the dad. So we'll look at this together in class too, to help it make a little bit more sense. But electrophoresis is a really cool process that we use to analyze DNA.